In today's video, we're going to run through the 4 key food tests that you need to master the GCSE biology exams. Now, our first objective is to be able to describe how to carry out the 4 food tests. The second objective is to know the positive result for each test. But first of all, before we can actually test the food, we need to prepare a sample. So you'll be using a pestle and mortar, and as you can see in the diagram at the bottom left, the pestle is kind of the rod that's used to grind the substance, and the mortar is the kind of container that you put the food in. So first of all, you grind your food using a pestle and mortar, whether it's pasta or crisps or something of that nature. Secondly, you add this to a beaker with distilled water. Third of all, you're going to stir that mixture with a glass rod. So then you should have a nice mixture of the food and the water. Finally, you're going to filter this using a funnel and filter paper so that you've taken out the solid parts of the food and you're just left with a solution. Now, the first test is the Benedict's test for sugars, or known as reducing sugars. Now, first of all, you're going to add around 5 centimeters cubed of your food sample to a test tube and place that in a water bath at around 75 degrees, but mark schemes will often allow 65 degrees and up. Next of all, you'll add 10 drops of Benedict's solution to this. Give it some time in the water bath and a positive result will be green, yellow or brick red. Green will be a low amount and brick red will be a high amount of sugar. Next of all, using iodine to test for starch. This is a fantastic little experiment. First of all, you're going to add 5 centimeters cubed of your food sample to a test tube. Then 3 drops of iodine and shake it gently. And the positive result for this one, guys, is the starch should turn blue-black if it is present. If not, the iodine will remain brown. Okay, another food test here. We've got the Biuret test for protein. Now with this one, add around 2 centimeter cubed of your food sample to a test tube. But again, this is a qualitative test. The exact amounts don't matter, we're just looking for a colour change. Next, you're going to add around 2 centimeters cubed of your Biuret solution and shake it gently. Now the positive result for this one is purple. And I've got a nice little test tube there with a purple solution showing a positive result for Biuret. Our next test is the Emulsion test for lipids. There are different tests you could use for lipids. We're going to focus on this one. Now the first thing to do is to add your food sample to a test tube. After this, you'll add ethanol, then you'll add water, and it has to be in that order. And the positive result for lipids is a milky white emulsion, which will indicate their presence. Okay guys, now time for a little bit of exam practice. So the first question asks us to draw a line from the food molecule to the reagent found. And the second one asks us for a safety precaution, which I haven't mentioned yet, so let's see if you can get it right. Pause the video here guys, and let's see how you do. Time for the answers now. So for proteins, we will use Biuret reagent. For starch, we will use iodine, and for sugar, we'll use Benedict's. Now a safety precaution for using Benedict's will always be to wear goggles because it can harm your eyes. Another one could be to use a water bath so that you're not using, for example, a hot Bunsen burner where you could have a risk of burns. Right guys, that's all we've got time for today. Don't forget to like, comment, subscribe. Thank you very much. See you in the next one.